Hello everybody, my name is Yasmeen Taalab. I'm from Egypt. I'm an emergency physician and also I'm a forensic doctor working currently in Germany at the Institute of Forensic and Traffic Medicine, Heidelberg. I'm gonna present today the post-cardiac arrest prognostication. I'm gonna focus on the microRNAs as a novel biomarkers and plausible therapeutic targets after cardiac arrest. First things first, why we prognosticate. Actually, we prognosticate the outcome at an early stage after cardiac arrest to make us able to tailor the patient care. This could help us as a physician to take the decision or to triage the decision on whether to enhance therapeutic efforts in patients with predictable high morbidity or to readdress the overall management or ultimately to make decision on withdrawal of care. Even with successful resuscitation and return of a spontaneous circulation, post-cardiac arrest syndrome yet contributes to unfavorable neurological and cardiac outcome. This includes the hypoxic brain injury, heart dysfunction, systemic and ischemic reperfusion response. Unfortunately, there is still a lack of sensitive and specific biomarkers for easy identification of these patients, and therefore tremendous studies evaluated predictors of poor outcome after cardiac arrest, including the patient's characteristics, clinical settings of the cardiac arrest and resuscitation, neurological examination and neuroimaging, electrophysiological studies and biomarker, which is the focus of our presentation today. Due to heterogeneity of the cardiac arrest population and scenarios, no single factor has been identified as a reliable predictor of the outcome. Before digging more into our presentation, let me show you our diagram that shows the most commonly used biomarkers to assess the degree of the organ damage after cardiac arrest. The first circle on the top surrounds the brain. The neuron-specific enolases and the protein S100B are released into the cerebrospinal fluid and then into the systemic circulation after brain injury. GFAP and the brain-derived neurotrophic factor are newly prognostic circulating biomarkers of neurological outcome. The second circle in the middle surrounds the heart. It's for the troponin and the brain natriuretic peptides are the most commonly used biomarkers after cardiovascular injury. On the bottom, the third circle surrounds the peripheral circulation where a number of pro-inflammatory biomarkers are released into the blood after cardiac injury and contribute to the development and exaggeration of the sepsis-like syndrome. Here is the biomarkers, which is the C-reactive protein, procalcitonin, interleukins, and tumor necrotizing factor. In our previous study that we present as a poster presentation in the ERC last year, we present a new panel that could also be used as a neurological outcome predictors after cardiac arrest and return of spontaneous circulation. That is the ER, the endoplasmic reticulum stress-mediated apoptosis, beside the cytokines and the neuroinflammatory biomarkers. This study was conducted actually on animals. However, the currently available predictive methods and the biomarkers lack sufficient accuracy and therefore cannot be generally recommended in the clinical practice. We are not using them yet in the centers that deal with the cardiac arrest or trauma centers. Why? Actually, we could explain this by confounders and the influence of the blood-brain barrier. The confounders. The idea of interpreting data from the biomarker studies is confounded by different factors, making it difficult to compare studies in a systematic review and in the meta-analysis in a meaningful way. These confounders could include the differences in the study design, inclusion and exclusion criteria of the patient, duration of treatment, the time of sampling, the laboratory evaluation, and differences in evaluated endpoints of the treatment. And that's why it's always in infinity a further large-scale studies are needed with accurate methodology, and therefore there is an urgent need for identifying a novel biomarker that can guide patient management at an early stage after cardiac arrest. Now the second cause, which is the influence of the blood-brain barrier integrity on the brain protein biomarkers clearance. The brain-enriched proteins of the tissue, that is, biomarkers, so the tissue fate, what's happened in the tissue, finally end with proteins, and these proteins are the biomarkers that are increased in the body fluids following the brain injury and cardiac arrest. The two most studied protein biomarkers include the primary astrocytic S100 calcium binding protein B, that is known as S100B, and the neuronal neuron specific enolase. Even though neither is protein specific, neither is brain specific. Both are highly brain enriched and have been associated with a worse intracranial condition and long-term functional outcome following traumatic brain injury, for example, but neither are brain specific. In fact, some centers using them in the clinical routine work, hence making these two biomarkers a particular interest to the neurotrauma translational research field. Here is some explanation about the blood-brain barrier integrity. And here we explain that the adult central nervous system comprises three anatomical barriers actually, namely the brain cerebrospinal barrier, the blood-brain barrier, and the meningeal barrier. For simplicity and to align with the clinical research field, we refer all of these barriers to blood-brain barrier. In addition, a transport route denoted lymphatic circulatory system has been recently described in experimental models. Although it has been suggested in preclinical models that the brain-enriched protein and the CSF passively leave the brain through the perivascular lymphatic circulatory system, others claim that the blood-brain barrier integrity plays a more decisive role. In traumatic brain injury, for example, in which the patient likely has concomitant injuries to multiple CNS barriers, the gold standard strategy for assessing the blood-brain integrity and disruption is the CSF to blood albumin quotient. Previous studies of the traumatic brain injury and subarachnoid hemorrhage not shown any significant correlation. However, these studies have been limited by the lack of longitudinal analysis and high frequency sampling during constant CSF drainage. So our research question is, what's happened in facts and in actual fact inside the brain, and what kind of novel biomarkers to be used? We started a collaboration between our institute, that is the Institute of Forensic and Traffic Medicine, together with IMBIL. IMBIL is a European molecular biology lab. So we started autopsy research. This is piecing the puzzle of the cardiac arrest and related death. The idea of autopsy as a research is not an entirely novel concept, but a key factor in conducting post-mortem research is the quality of the tissue. Unlike animal tissue, whose condition at death can be controlled and influenced, the human tissue can only be controlled naturalistically. This introduces potential confounds based on pre and post mortem condition that may influence the quality of the tissue and its ability to yield accurate results. The traditionally recognized confounds that reduce the tissue quality are agonal factors, coma, hypoxia, hyperpyrexia at the time of death, and long-term post-mortem interval. We measured the tissue quality parameters in over 20 post-mortem cases controlled from different sources and in 20 multiple different brain regions. Our results show that the most sensible indicator of the tissue quality is the RNA integrity number. Moreover, even when RNA was degraded, the protein level remained stable. However, these correlations did not prove true under all circumstances. For example, surgical tissue that yielded unexpected quality indicator. These data suggest that cases whose source was a medical examiner's office represent a high tissue quality. Till the moment I'm presenting now, we didn't publish yet our results, but we succeed with one PhD thesis entitled Optimization of a Method for Extraction and Analysis of Chromatin from Human Brain Tissue Conserved in Formalin over various periods of time. We have an overview of our work that starts from the autopsy and then dissection of the brain, and then taking these tiny brain regions into histocassettes and then keeping them into formalin and then doing the molecular analysis. Then we have also these kinds of percentage of the yield of the DNA, RNA, and the quality of the DNA and RNA as well, to make sure that we have the best quality of the results. But I'm not gonna go through this more because we are in the stage of writing the manuscript and publication and so on. Why we choose the biomarkers and why we choose the microRNA. So we now goes with autopsy research using a post-mortem human brain tissue, and then with a microRNA for further study about the cardiac arrest, traumatic brain injuries, and neurodegenerative diseases. So why microRNA? MicroRNA are short, non-protein coding RNA molecules that are evolutionarily conserved and ubiquitously expressed. Similarly, microRNA are abundantly expressed in the brain, where they play a key role in development, plasticity, and disease development. Significantly for the biomarker research, circulating microRNA are stable, that is protected from the RNA's degradation, and can be easily investigated using conventional PCR methods. We succeeded to publish a review article focusing on the microRNA expression profile as a proposed mechanism for disease development. As I said before, for traumatic brain injuries, for the consequences after cardiac arrest, and the neurological outcome for the neurodegenerative diseases and traumatic brain injuries as well. Our take-home messages. The only way to know what's going on with the brain, in which the situation is very complex, is to go through the human brain tissue. So we conducted this autopsy research using human brain tissue, and we are using more stable, conserved biomarkers in our approach for the best understanding for what's happening in the cardiac arrest and the events and consequences. Also to know more about the mechanism of different diseases like traumatic brain injuries and neurodegenerative diseases. I hope you have a valuable time with my presentation. Your feedback is very valuable to me, and I wish you a very valuable time in the 2020 ERCE Virtual Congress. Thank you so much.